Consider this clinical presentation for a moment. A 38-year-old woman comes in with a 15-year history of gradual weight gain. She currently weighs 468 pounds. Her blood pressure is sitting at 150 over 100 millimeters of mercury, and her fasting blood glucose, it's at 180 milligrams per deciliter. That is way above the normal range of 60 to 100. For the clinical physicians and public health scientists joining us today, this exact scenario is becoming incredibly and alarmingly common, isn't it? But did you know the geographic variability of this disease is absolutely staggering? We are looking at a 400-fold difference in the likelihood of developing type 1 diabetes between a child in Finland, where the incidence is the highest, and a child in China, where it is the lowest. The epidemiological stakes we're dealing with here are simply massive. Okay, let's dive right into this. Welcome to today's explainer on diabetes and clinical management. Whether you're a food technologist, a clinical endocrinologist, a dietitian, a toxicologist, or a biochemist, the mechanisms of diabetes intersect directly with your daily professional work. We're going to unpack this complex disease together, starting from the clinical baseline all the way down to the molecular cascade. Our agenda today follows a precise clinical to molecular structure. 1. The clinical reality. 2. Mechanisms of insulin action. 3. Novel hormonal mediators. 4. Navigating metabolic complications. And 5. Clinical and nutritional management. Let's begin with Section 1, the clinical reality, understanding the baseline. Now, it's absolutely crucial to understand the stark contrast in etiology here. Type 1 diabetes involves very low or sometimes completely absent plasma insulin, but it accounts for less than 6% of cases. Type 2, on the other hand, accounts for about 95% of all diabetics in the United States, and crucially, it doesn't begin with an absolute deficiency of insulin. Actually, it starts with cellular resistance. The tissues simply decrease their ability to respond to normal, circulating amounts of insulin. Which brings us to Section 2, mechanisms of insulin action, the cellular engine. For our biochemist and analytical chemists, think of the insulin receptor as a locked glycoprotein gate, sitting right there on the plasma membrane. It's a highly complex structure. We have a heterotetramer made of 2 alpha subunits and 2 beta subunits. The entirely extracellular alpha subunits act precisely like a keyhole, just waiting for that insulin molecule to arrive. Now, when insulin, our key, binds to the extracellular alpha subunits, it unlocks the gate. This binding stimulates the tyrosine kinase activity of the transmembrane beta subunits. This results in autophosphorylation, which initiates a highly complex intracellular phosphorylation cascade. With strict scientific rigor, we can confidently say this specific kinase engine is what ultimately allows glucose uptake into your muscle and adipose tissues. When this very cascade fails, well, that is when we see insulin resistance. Let's expand our view in Section 3. Novel hormonal mediators, beyond insulin. We'll start with adiponectin. This is a protein hormone secreted directly from adipose tissue. But here is a fascinating biological paradox. You would naturally expect more fat tissue to produce more of this hormone, right? Yet in obese individuals, adiponectin concentrations are actually reduced. When a patient successfully undergoes weight reduction, their circulating adiponectin levels increase, and that subsequently increases their insulin sensitivity. It's quite remarkable. Next, let's bring our dietitians and clinicians into the fold with bone biology. Did you know that osteocalcin, the second most abundant protein in our bones, actually acts as a hormone? It regulates blood glucose, influences pancreatic beta cell function, and even increases the secretion of adiponectin. Because building bone mass through weight-bearing exercise naturally increases osteocalcin, exercise directly boosts insulin production and sensitivity, quite literally from the skeleton outwards. From a pharmacological perspective, phyto A presents a brilliant target. It's produced in the liver, and its traditional role is keeping calcium and phosphorus solubilized in serum. However, it also acts as an endogenous inhibitor of the exact insulin receptor tyrosine kinase we discussed just a moment ago. High baseline levels of phyto A are a remarkably strong, independent predictor of developing diabetes. So disabling its action on the insulin receptor without disturbing blood calcium, that is a major target for future drug development. Finally, we really must connect circadian rhythms to public health science. Melatonin receptors, specifically coded by the MTNR1B gene, are actually present right there on human pancreatic beta cells. When melatonin levels are high at night, insulin production directly goes down.
This perfectly explains why population studies consistently show that getting less than 5 hours of sleep a night significantly increases the risk of type 2 diabetes. Chronic sleep deprivation severely disrupts this delicate glucose regulation.
Moving on to Section 4, Navigating Metabolic Complications, the Toxicological Fallout. So, what happens to excess glucose inside tissues that don't require insulin for uptake, like the ocular lens and our peripheral nerves? Elevated intracellular glucose, catalyzed by the enzyme aldose reductant, forms sorbitol. Because sorbitol diffuses very poorly across cell membranes, it basically gets trapped and accumulates intracellularly. For our toxicologists, this causes immense osmosis-induced disturbances, leading directly to structural damage like cataract formation and peripheral neuropathy. Systemically, this metabolic breakdown is incredibly severe. Without effective insulin action, carbohydrate metabolism fails entirely, causing hyperglycemia. Protein catabolism increases, leading to urinary nitrogen loss. And crucially, without insulin to inhibit hormone-sensitive lipase, fats break down rapidly. This floods the system with acetyl-CoA, which quickly converts into ketone bodies, forcing the patient's body into a highly dangerous state of ketosis and acidosis. Let's look at how we fix this in Section 5. Clinical and nutritional management, reversing the trend. The good news is we have evidence-based interventions. Randomized controlled trials in the US and Finland deeply highlight the cascading benefits of lifestyle intervention. The targets are highly specific yet achievable. Achieve a weight reduction of 5% or more, exercise moderately for at least 30 minutes daily, and increase fiber intake. The results are profound. Achieving just a mere 5% weight loss alongside these basic steps results in a massive 58% reduction in the risk of developing diabetes. When translating these goals into food technology and everyday dietary strategies, there are clear parameters. While 55 to 60% of calories should come from carbohydrates, they absolutely must be complex carbohydrates. Sucrose should be strictly limited to 10% of meal calories. Interestingly, fructose produces only 20% of the glycemic response of glucose, making it a potentially advantageous sweetener for food technologists to utilize. And targeting 40 grams per day of dietary fiber is absolutely critical for reducing that insulin resistance. Let's see how this builds into alternative medicine compounds, which offer truly fascinating molecular mechanisms. Ginepin, derived from gardenia fruit extract, actually blocks the UCP2 enzyme, which otherwise inhibits insulin secretion. Then we have bitter melon, which stimulates the AMPK enzyme. This effectively mimics the molecular effects of exercise to increase glucose uptake. And even simple household vinegar, just 2 tablespoons before a meal, has been shown to reduce post-meal blood glucose by 20% in type 1 patients. These compounds are an absolute goldmine for pharmacological research. We have journeyed all the way from stark clinical vitals to the cellular gatekeepers of insulin, and through the highly complex roles of bone proteins, sleep hormones, and botanical compounds. The science across all these disciplines is deeply interconnected. So, I leave you with this provocative thought. As we continue to uncover these incredibly diverse molecular secrets, how will you and your specific discipline integrate these novel pathways to reshape the future of diabetes management? Thank you for joining this explainer.